This presentation is about the changes of insulin resistance and beta cell function in women with gestational diabetes mellitus and normal pregnant women during mid and late pregnancy. Mid and late pregnancy start from 20 weeks to 24 weeks. The purpose of this study was to observe how insulin resistance changes and how beta cells function during mid-pregnancy in women with GDM. Basically, GDM is a condition which women without previously diagnosed with diabetes exhibit high blood pressure during pregnancy. Basically, 64 pregnant women having parental care underwent an oral glucose tolerance test, which is performed to screen for gestational diabetes. This test involves quickly drinking a sweetened liquid which contains a 50 gram of glucose. The body absorbs this glucose rapidly, using blood glucose levels to rise within 30 to 60 minutes. A blood sample is taken from the vein in your arm about 60 minutes after drinking the solution. The blood test measures how the glucose solution was metabolized, which is how it was processed by the body. Also, an insulin release test was taken at 20 to 24 weeks of the pregnancy that can be explained as insulin and C-peptide levels may be used to monitor the amount of insulin produced by the body, called the endogenous insulin, to check if the body is not responding to insulin properly, which is called the insulin resistance test. From the group of 64 pregnant women, 34 were diagnosed with GDM, gestational diabetes mellitus. The other 30 pregnant women with normal blood glucose were the control group, and a control group are the patients who received either no treatment or a standard treatment. Changes in the insulin resistance and beta cell functions were shown with the oral glucose tolerance test and the insulin release test, which were both explained. During the 24 weeks of the pregnancy, the insulin resistance and beta cell function was much higher compared to pregnant women in their mid-pregnancy, which is about 20 weeks for both groups. People with gestational diabetes mellitus had a lower insulin secretion index in both mid and late stages of pregnancy. The values of the area under the curve that we will see in the next slide will show that blood glucose level in the mid and late pregnancy were higher for GDM group compared to those in the control group, and the insulin resistance level was much higher in GDM patients than in the normal pregnant women.
This graph shows the blood glucose level for pregnant women diagnosed with GDM. In 20 weeks of pregnancy, the blood glucose level is about normal, which is about 160 mg per dl. As they go further with the pregnancy, the blood glucose level goes higher and reach to a very high level of 180 mg per dl when the woman is 24 weeks pregnant. This experiment shows that the insulin resistance increased as the gestational week increased in women with GDM and also with the normal pregnant women. The beta cell's ability to fulfill the increased insulin resistance was aggravated by modulating insulin secretion. Pregnant women with normal metabolism of glucose had a lower insulin resistance compared to women with GDM.